This is a novel bispecific antibody. The other name for it is Cevostamab. And it's targeting a new target in multiple myeloma called FcRH5. And this is a cell surface receptor that's expressed only in the B cell lineage and in no other normal cells in the body. So it's on B cells at a low level. It's expressed higher on plasma cells and particularly high on malignant plasma cells in myeloma patients. And so this makes it an attractive target for immune therapy. And so Cevostamab is a bispecific antibody. One end of the molecule targets FcRH5 on the myeloma cell. The other end targets CD3 on T cells. And this leads to T cell activation within the bone marrow microenvironment, eventually leading to myeloma cell killing. And so after this molecule demonstrated preclinical activity, this phase 1 first-in-human dose escalation study was opened, and we're presenting the first data from this trial. The study is in relapsed refractory myeloma patients that have exhausted sort of all available options. It was a heavily pretreated population of patients with a median of 6 prior lines of therapy. 72% were triple-class refractory, and 45% were we'll call pentadrug refractory, meaning they're refractory to 2 proteasome inhibitors, 2 IMiDs, and a CD38 antibody. Almost all patients were refractory or progressing on their most recent line of therapy, and interestingly, 21% had a prior BCMA-targeted therapy, either CAR T cells or an antibody-drug conjugate. The study was set up in a typical 3+3 dose escalation design. However, there are some interesting tweaks to it. And so one of the known side effects of bispecific antibodies is cytokine release syndrome. And so in order to mitigate this, the current cohort, which we were presenting at ASH this year, engaged in what's called single step-up dosing, in which patients received a low dose of the drug on cycle 1, day 1, and then if they tolerated that, they got the full dose starting a week later on day 8. And then subsequently, they would receive the target dose once every 3 weeks, up to 17 cycles for disease progression or unacceptable toxicity. The drug's given as an IV infusion. Patients were hospitalized for the first 2 doses up to 72 hours to monitor for CRS or other toxicities, but then they could receive the drug as an outpatient thereafter. And the primary objective is safety and tolerability and identification of the recommended phase 2 dose. So what we presented at ASH this year was the initial safety and efficacy data for the first 53 patients who were available at the time of data cutoff who were enrolled into this single step-up cohort. And in terms of toxicity, we did see hematologic toxicity, namely anemia, neutropenia, thrombocytopenia. This was grade 3 or 4, roughly 15 to 25% of patients. The most common toxicity was cytokine release syndrome. This was seen in 76% of the patients. However, all but 1 patient had a grade 1 or grade 2 CRS. There was only 1 grade 3 CRS event. Almost all the CRS occurred during this cycle 1, day 1 step-up dose or day 8 dose, and it was very uncommon at subsequent doses. Neurotoxicity was seen in roughly 28% of patients. This also tended to occur during the CRS and was short-lived and self-limited in the majority of patients. 23% got tocilizumab and 17% got steroids. And importantly, the step-up dose, which eventually got fixed at 3.6 milligrams, allowed us to subsequently escalate the target dose, and dose escalation is still ongoing for that dose at 160 milligrams and climbing. And so by giving the step-up dose, we've been able to escalate the target dose up without an increase in the frequency or severity of CRS. In terms of efficacy, we did not see any activity at lower doses of 10.8 milligram target dose or below. But starting at 20 milligrams and above, which are considered the active doses of the drug, 34 patients were enrolled at those active doses, and 18 of them, or 53%, had an objective response, including 32% with VGPR or better and 18% with complete remission or better. This did include even MRD negative CRs in a subset of patients that were evaluable for this, and included responses in pentadrug refractory patients, as well as in patients who had progressed on prior BCMA CARs or antibody-drug conjugates. So the escalation is continuing. We still don't know the recommended phase 2 dose yet, but clearly seeing some activity, particularly at the the highest dose levels, 90 or 132 milligrams, where the response rate was 61% in that group so far. Durability of response, I think, is still a little bit early, particularly at the higher dose levels, but we do have a number of patients who have durable remissions longer than a year, including some who are ongoing even after stopping therapy. So I think we just have to wait and see what the median duration of remission is going to be. So I think the take-home messages from this abstract are, number one, that FcRH5 is a viable target for myeloma. This is the first study really showing high response rates with a drug targeting FcRH5, and in particular Cevostamab, this bispecific antibody, which has very convenient dosing once every 3 weeks, has activity in a highly refractory group of patients. The main toxicities are CRS, and to a lesser degree neurotoxicity, which are expected, but largely manageable and largely occur within the first cycle. And where the study's going now is continuing dose escalation at the single step-up dosing. Studies also initiated a double step-up dosing cohort where they get even a lower dose on day 1, sort of a medium dose on day 8, and then the full dose on day 15 to see if that further decreases the incidence of CRS. And so both of those are currently accruing, and then there will be expansion cohorts as well once we sort of get to the recommended phase 2 dose to really get a better sense of efficacy and safety and tolerability once that dose is established.